Good afternoon, everyone, and welcome to the webinar, today's webinar. My name is Lord Abbey, and today I'm coming to talk about increasing food and nutrition security while sustaining soil ecosystem services. We know food, especially plant food, comes from the soil. And even if we're talking about animals' food, the source is from the soil. So, we need to have good soil health, so that we have good food for humans, good food for our livestock, so that we can have good food and health overall. And I'm talking from Dalhousie University and Department of Plant Food and Environmental Sciences today. Okay. So world population, as we all know, is around it's going to be around 9.7 billion by 2050. It's increasing at an astronomical rate. And therefore, we have to increase our food. If crop yield does not increase by 60% by 2050, we'll run into a big problem. And even now, due to the sustained global climate change, we are feeling it now, where food are various foods are in short supply. So this is a serious problem. As a result, our farmers, due to pressure from the policymakers, pressure from consumers, pressure from the society, have to do intensive crop production. And doing intensive crop production implies using the same size of land and the same soil resources. It means we have to do something to the soil. They have to do something to the soil because the soil is also running out of its resources that support plant health and productivity. And how do they do this? Most of them rely heavily on synthetic chemical fertilizers, which we all know is not very healthy. And this has caused various environmental stresses to crop production. The current trend that everyone is yearning for is to reduce this synthetic chemical fertilizer, the use of this synthetic chemical fertilizer in our agricultural soils, so that we can sustain, the soil health and crop productivity and human health as well. The alternative strategy that stimulates plant productivity and resilience, can come from various sources. So we don't need to rely on these synthetic chemical fertilizers, but we have to be smart and to ensure sustainability. Globally, the cost of solid waste management in 2010 was $205 billion US. Now, why solid waste? Why is it interesting? Or why do we even have to talk about solid waste? Solid waste, especially from food and our kitchen, restaurants, etc., contains nutrients, very important nutrients that can be recycled and reused. That's why we're talking about solid waste here. And as I've said already, the cost of solid management of this solid waste, because if you want to manage it so that otherwise you're leaving it around, it can cause it's a health hazard. The cost is about 205 billion, as I've said already. But it's going to increase. It's going to increase because our lifestyle has changed. People are eating more, people are wasting more, and population is increasing. So it's going to change. And for that matter, the World Bank estimated that in 2025, the solid waste management is going to cost about $275 billion US. That's no joke. The good news is that 30 to 64% of these total municipal solid waste constitute biodegradable organic materials, which is, as I've said already, can be composted because they contain nutrients and other non-nutrient bioactive compounds that the plant can benefit from. Compost is generally rich in beneficial microbiome. I don't have to overstress this because it's well known. And it's also rich in humic and non-humic substances. So if you're talking about fulvic acid, you're talking about humic acid, you're talking about all humins, these are all in can be found in compost. Compost is rich in that. If you're talking about nutrient elements, if you're talking about some proteins, amino acids, some Other compounds, they are all present in compost that can, including plant growth hormones, they are all present in compost that plants can benefit from. Therefore, composting offers an immediate solution to the problem of wasting organic materials for recycling soil organic matter and soil nutrients needed for plant growth. So we don't have to waste these, materials, the municipal solid waste that I'm talking about. We should not waste it. We should rather compost it so that we can use that to enhance soil health, enhance crop productivity, and so on and so on and so forth. Therefore, this particular project was started to support the advancement of CQA. CQA means Compost Quality Assurance, tested municipal solid compost market in Manitoba and the whole of Canada. And CQA is a set of rules or regulations or standards that was developed under the auspices of the Compost Council of Canada to ensure that compost that is produced from municipal solid waste meets certain standards, so that they will not negatively impact the soil. Because compost, if it's not produced in a certain way and doesn't have certain qualities, can be devastating, can be problematic. So we need to have a compost that has quality standards that it will benefit the soil as well as the crops. So the specific objectives of the project was to determine the application frequency of CQA tested municipal solid waste compost on soil quality, plant growth, harvest quality, and economic benefit. So it's a broad spectrum of specific objectives that we're looking at. So as I've said, it's 5 years we did this work in Brandon, Manitoba, and it's organic farm. They've never used synthetic chemicals for since we started the first time we started was, about 30, 40 years, no synthetic chemical fertilizer applied on that farm. It's all organic all the time. One thing, the climate there, in Brandon is a cool to moderate cool boreal subhumid continental climate. So they have very short growing season. It means you have to start your crop early at the right time, and then also finish it early at the right time. So we have a short frost-free days that is cropped in that particular area. The soils are sandy loam, they are quite light, and they are rich in calcium as well, and also some organic matter. However, the soil that we grow our crops on were depleted because they were continuously cropped, without use of synthetic chemical fertilizer. Less compost was used there. They were continuously and intensively cropped. So we looked for a depleted soil. That's where we started our experiment from. So that's the classification for you. It says orthic black chenosim, rich in organic matter, that's the soil type. And the compost was also obtained from the city of Brandon. They supply us the compost. They follow the CQA standard regulations to produce those compost, the compost that we used. We did lettuce, we did beets, we did green beans. So we do this every year. And the plants are, cultivated in rows. We have 3 replications. We use randomized complete block design and with 3 replications and so on and so forth. The treatment are that we have plots that did not receive compost at all. So no compost application. And then we have plots that received compost every year. And then the third plot or treatment, we apply compost every 2 years. So we have no compost, annual compost application, and biennial compost application. This continued for 5 years. And the no compost was on the same, all the treatment were on the same plot throughout the 5 years. So we don't change the plot, same. However, we try to shift or move the plants. If we grew the same plant, but not the same plant at the same spot every year. No, we try to sort of rotate them within the plot. We collected data on soil, so soil physiochemical component. We collected soils on soil for microbiome analysis, and also we did functional profile of bacterial community, because when we did our soil microbiome analysis, we realized that the fungal population were similar. So we rather focus on the functional profile of bacterial community, and also we did plant growth and yield analysis. So the soil physiochemical properties, so if you look at this table, you can see that with the water saturation capacity, look at the field capacity, you can see that they were all increased in the annual application compared to the control. Similarly, the biennial also increased more than the control. But if you compare the annual and the biennial, you can see that the annual did very well, better than the control. So we're able to increase water retention or water holding capacity of plots that received or soils that received annual application of compost, more than the biennial, and the biennial was more than the control. If you look at the wilting capacity, so this, when the soil is depleted, you look at the wilting capacity, you can see that even the annual and biennial were able to hold much, a bit higher, slightly higher than the control. Okay. And then we have the soil, the bulk density, the particle density. If you look at the bulk density, because of the organic matter increase, you can see that the bulk density of the annual was reduced, followed by the biennial. And then if you look at the control, you see most soils on the average have a bulk density of 1.2 grams per centimeter cube, so per cubic centimeters. So this range falls within documented values for soils in general. So, so you can see this proves also, or can demonstrate that the addition of the compost actually reduced the bulk density because of the organic matter content, increase in organic matter content. And you can see also from the water holding capacity, which can also be attributed to the increase in organic matter content, as we shall see from here. So electric conductivity and then the turbidity, these are all water quality parameters that we measured from the soil. And as you can see, the annual had higher EC, which is the electric conductivity, compared to the biennial, and then the least was in the control. Same things applies to the turbidity. The turbidity will tell you about the dissolved organic matter in the soil, while the electric conductivity will give you an idea of the nutrient content in general. It's not discriminatory, so it will tell you the general concentration of nutrients in the soil. You can see that the annual far was higher than the biennial. If you look at it, over 2,000 micro cement per centimeter. And if you look at the biennial, you can see 715, and look at the control, it's about 377. So the annual application really increased the EC. If you look at the organic matter content too, it's proven similarly. Look at the total dissolved solids, and then look at the, oh, sorry, my fault, I have the EC twice, but if you look at the EC, the organic matter, the total dissolved solids and the salinity, you can see that it's very, very high in the annual compared to the biennial, and then the lower or the lowest was in the control soils. So I'll go back one. So this tells you that, actually by adding the municipal solid compost, as we all expect annually, it really, really, really did great job compared to adding every 2 years. Because some people are afraid that when you add municipal solid compost, it affects the soil, adds some compounds, chemicals, and so on and so forth. It's true, it has some element, but those elements have been proven to not to be toxic to the soil based on our previous publication. So this is a good news. And this is very encouraging. So let's look at more into the soil nutrients. So we have the organic matter here plotted against years. So the 5 years of the trial. And if you look at panel A, you can see that year 1, year 2, year 3, year 4, you can see the black line, which is the annual application, and which is talking about the soil organic matter. You can see that it keeps on increasing, okay, compared to the, right from year 1 when we started increasing, and then compared to the biennial and then the annual, which is expected because we are adding compost, so we expect that to happen. Now, if you look at the saturation phosphorus level, the same thing. You look at the potassium to magnesium ratio, the same thing, which is interesting because in sandy soils we expect them to have very low potassium levels. So if addition of the compost is able to increase it to a level that the plant would benefit, then it's good. And we can see that it leveled off after year 3 because whatever is being added, the plants are taking it up, and therefore, it's keeping the concentration constant throughout the year. If you look at the pH, so the pH increase in the annual, okay, it increased in the annual, it increased in the biennial, and then leveled off. This is because once we are adding the compost, we are increasing the soil organic matter content. And we know organic matter added to the soil has buffering capacity against pH. So it will not allow the soil acidity to increase above certain level. You can see that it has some buffering effect that is controlling. So it increased but started declining. And this could also be due to increased calcium. So the basic ions, increased calcium content of the soil. Then we have CEC, which is cation exchange capacity, and you can see the cation exchange capacity. It increased generally from year 1. It increased. If you look at it, it increased, but it's similar. There's no statistically significant difference among the 3 soils. And this is because it seems that whatever is being added, because if you look at the previous graphs, you can see that the cations, potassium, magnesium are being increased. So it looks like whatever is added, the plants are able to take it up. And then when they take it up, the level becomes similar to the control, which is fair enough. You add, the plant is able to take it up. And moreover, we know that compost is a slow release. It's a slowly releasing in terms of nutrients to the soil. So after adding whatever is the compost is able to release, the plants are able to take it up. And therefore, it's keeping the CEC fairly constant from year 2. That's what we are saying. It fluctuates, but not to any significant level. So we don't see any significant differences. If you look at the calcium to magnesium ratio to the same, you can see that it reduced from year 1, it reduced and then it climbed again. So I don't know why it reduced, it declined. Probably we added a compost in year 1, we grew our plant, so maybe the plant took more of this element from the soil. Therefore, the calcium to magnesium ratio declined. Maybe we're having more magnesium there from the compost, so it declined, and then adding, it climbed up again. So you can see that the control soil had more calcium because maybe the magnesium is being used up, by the plant. So you can see it has, because that soil, the soil by nature has a lot of calcium in it. It's rich in calcium. That's the nature of that particular type of soil, the orthic type of soil. It's rich in calcium. So you can see that the control here has a lot of calcium in it compared to where you have where we're adding the compost, which also brings in the magnesium. So if you don't, then you have higher calcium. With the magnesium, you can see that it's declined a bit, because the magnesium level was high. So the ratio declined a bit, which is acceptable. If you look at the estimated nitrogen release, which is the nitrogen available for the plant to take up in that particular season, you can see that the annual application had more of the available nitrogen, followed by the biennial, and then less in the control soil. In fact, in the control soil, after year 3, it started falling because we are not adding anything to it. Whereas if you look at the biennial and the annual, it remained again like it plateaued because we are adding, the plant is benefiting at the same time. And we can see that from the plant growth response. We can see that from that. Okay. Exchangeable sodium increase, of course, because it's municipal solid waste. We expect the sodium to be high. So again, the exchangeable sodium increased in the annual, but it wasn't detrimental because and the plant response does not show any toxicity from sodium. So it increased in the annual, followed by the biennial, and then less in the control plants. So that's the for the physiochemical. Now, when we come to the microbial, the microbiome analysis, what we found that, so the 16S is for bacteria, and then the ITS is for fungi. And you can see that the evenness and then the diversity of microbes, and so on. So the evenness, we talk about the distribution and so on and so forth. You can see that if you look at the control compared to the biennial, compared to the annual, you can see the differences there. Okay, so the control has less, whereas we have more in the biennial and then the annual. That's for the bacteria. If you look at the evenness, again, you can see that, well, it's statistically not significant for the evenness. And then if you look at the diversity, you can see that the diversity increased more in the biennial and then the annual compost. We expect this because we are adding compost, so we expect that. Yes, if you look at the fungi generally was not significant, different, because you can see that they all have, except the observed features that you can have, we had more in the biennial and then in the annual than the control. But generally, the fungi microbiome did not change much significant compared to the bacteria. And because of that, we focus on the bacteria profile more, the functional profile of the bacteria community more. And you can see, if you look at the panel A, you can see that the red dots are for the annual, the green for the biennial, and then the blue for the control. And you can see that there are separations, right? Except the biennial, which overlaps the annual and overlaps the control as well, because its biennial is medium. So you can see that, that is medium. You can see where it lies on the chart, on the chart. Now, if you look at the panel B, okay, look at the panel B, you can see that we had more functional bacteria profile in the annual generally compared to the biennial, followed by the biennial, and then less in the compost. If you look at the bar chart, so the black bar chart, you can see for each of the segments, okay, you can see that, it's higher in the annual than the biennial and than the control. And some of these microbiomes are there for energy. Some are there to increase metabolism of the bacteria itself. Some are there to add to enable the bacteria to do more mineralization and so on and so forth. So when you come to the plant productivity, you can also see, very obvious, that I have the carrot here because we did the carrot on the side and it did very well, but we didn't include it. We didn't include the data because at the end, when we want to do our final harvest, we had problem with that, so we didn't include it here. Now, when you look at the plant, you can see that there was clear difference, very highly significant differences in the productivity. Look at the annual, biennial, and the control. It was very consistent. When you enter the field, no one will tell you. You can differentiate by even looking. So, it was very clear that the distinction was very, very, very clear. So let's look at some of the components of plant growth and yield. Now, if you come to the shoot, we measured plant height. And with the plant height, we found in the annual that for green beans, for instance, we found that the annual gave higher plant height. So the plants were taller, looking more lush, more beautiful in the annual compared to the biennial and then the control. For lettuce, same. Those that received the compost were greener and more lush and beautiful than the control. Same applies also to the beets. We look at the leaf area. The leaf area too, we have this similar observation. So the leaf area and then the plant height, as I said, the annual was greater than the biennial, followed by the control. And this tells you that applying annual, compost annually to the soil increases productivity way more, or plant growth way more than the biennial. And then the control, at least you need to add something to get something. Then we look at the leaf dry matter content. The leaf dry matter content basically is an indication of how the plant is able to survive. It's a survivor because the dry matter content is carbon. If a plant is under stress, the plant tries to produce more carbon. So when you measure the dry matter content, it will be higher than the plant that is happier, because the plant that is happier is just channeling its energy into growing and producing flowers, producing more tissues and looking lush and beautiful, whereas the plant that is stressed tries to harden itself, build its skeletal structure so that it can survive. So a plant that is under stress tries to switch into a survival mode. And that's exactly what the leaf dry matter content was trying to measure here. So you can look, if you look at the control plant where the plants were struggling, they didn't have enough nutrients and everything wasn't sufficient, you can see that they had higher leaf dry matter content because they switched into surviving mode. So they were just, hardening their skeletal structure, if you may, just for them to survive. So that's what we can see here is stress adaptation. So they were under stress adaptation. So they had higher leaf dry matter content compared to the annual and then the biennial. So more for plant growth component, if you look at the stem, so we have specific stem diameter. So this measures the stem diameter. And again, the different plant responded differently. So you can see the biennial, the green beans for the biennial, it has higher stem specific stem density compared to the other treatment, and so on and so forth. If you look at the roots, the root one is very interesting because if you look at the green beans, you can see that the root in the control is higher, it's deeper. This is because those roots were in search of nutrient. So they have to grow deeper and extensively just to make sure that they get something to support the plant. And that's what we are seeing here. For lettuce has adventitious roots. And lettuce, they grow, they are luxury consumers. So once they are happier wherever they are, they try to grow and they try to be okay. So that's what we are also seeing here. And then the beets, with the beets, the annual did not, because the beets, how the beet forms, okay? It forms by what? Swollen stem. So, or swollen root, yeah. So it's modified its stem, and then when it's modified, it swells, it takes in more nutrient, water, and everything, and therefore the main root does not grow any further because it doesn't need it. It doesn't need to grow because it's all modified into the bulb that we harvest, and that's what is happening here. So we have, it forming, and therefore it's forming, and therefore there's no need for it to grow extensively or anywhere. So it's, because it's happy, it's fine, it's reaching maturity very early, and so on and so forth. But if you look at the picture, you can see that, it doesn't have to, because everything went into the bulb that we see here. So the root doesn't have to grow any extensively or too long to go anywhere. Whereas with the control, it has to, because it's looking for more nutrient, more water for the plant to be able to use. Okay, sorry, I went back too far. Oh, am I sorry about this? Okay, so that's what's happening there. Now let's look at the edible portion. So this is measuring the yield of the plant. And if you look at the yield, you in the annual for green beans, you have 434 kilograms per square meter, and then 414, and then you have 321. You can see the difference is highly, highly significant in the annual compared to the control. And same thing applies to the biennial compared to the control. And between the biennial and the annual, you can see that the annual is had higher yield compared to the biennial. Now let's look at the lettuce. The lettuce do the same. The lettuce, you can see comparing the annual to, even though statistically the annual and the biennial were not different, but to the farmer this is big because it's about 12 kilogram per square meter difference. So this is significant to the farmer who is growing on a large acreage, and then if you multiply this by the dollar, you can see that it's that's a lot of money. So it's significant. However, statistically, it's not. And so, and then you compare it to the control where no compost was applied, and it's only 10. There was nothing there. We saw that in the picture, right? Look at the bees too, the same thing. 311, that's 311, 260, and then 64. And we saw that in the picture as well. So when you go back and you look at the picture, you can see that, look at the control for the beets, look at the biennial and then the annual, and look at the leaves as well for the lettuce and then the green beans. So so the yield was highly significant, and this tells you that applying compost annually can get you the bags. Well, if you do your economic analysis, you have to do your economic analysis and know whether you should apply annually or biannually, because you can see that for all these, the green beans, the lettuce, they were not statistically significant difference. Okay, and then the beet was. So you have to do the math, how much is per unit compost, and where is the compost coming from, transportation, haulage, etc., etc. And then you can say, well, should I apply biannually or annually? But based on this research, I advise that compost should be applied annually to soils. We also did a PCA analysis, principal component analysis, just to bring everything together so that we can have look at the bigger picture and see what's really happening. So if you look at that, you can see these are the C is the control, no soil, no application. We have the biennial here, and we have the annual here. You can see that the treatments are well separated. They are well separated. They are not clustered at one point, but we have three clear, distinct clusters. We have that for the C control. We have that for the biennial, and we have that for the annual. So this tells you that the soils were very, very different after 5 years. They were highly different, and it's true. When you enter the field, you can just have when you look at the field, you can tell that this soil is different from this soil, is different from this soil. Okay, so what we measured and what I see here matches to with what is actually happening in the field. Now, what I also want you to look at is, you can see that the annual, you can see that all these components, so these are the functional components of the bacteria that we measured, the functional profile of the bacteria that we measured. And the elements here that you see, so the sulfur, iron, copper, phosphorus, magnesium, and so on and so forth that you see here are the soil elements that we also measured. You can see that the soil element can be associated with the functional profile of the bacteria. And you can associate that closely to the annual application of compost. What this means is that applying compost annually, you have more functional components of the bacteria, profiles of bacteria, which is very, very, very important for soil health. And then same time, you also have high, very rich, chemical elements, which we all expect. This followed by the biennial and then the C, because the C is very far from where these components are. So that's how you read this. Then again, we try to bring more things further. Okay, so we did so more things further. Again, you can see we have the lettuce here. So the previous one that I showed you, these are basically soil components. And then this here is soil and also plant components. So we have this lettuce. You can see the lettuce. So this is the control lettuce. This is the control green beans. This is the control beets. You can see where they are all clustered here, okay, on the left, far left. And then you can see the biennial here. You can see the beets here. And then you can see the biennial green beans here. They are also in the middle. And then when you come here, you can see the annual beets, the annual green beans, the annual lettuce on the far right. So you can see that here on the far right. So again, this matches with the soil classification that I showed you previously, where you have the control on the far left, you have the biennial in the middle, and then you have the annual on the far right. It matches with that, okay, similarly. And then again, if you look at the annual, they look at all the plants, the annual lettuce, the annual beet, the annual green beans, you can see that they are very tightly associated with the functional component, the functional profile of the bacteria. It means that having rich soil, having higher microbiome soil with functional profiles also has had impact on the plants, especially with the annual application of compost. Okay, that's why you have them here closer together, and then you have the biennial here, and then you have the control on the far side. The same thing, if you see P, those letters, uppercase letters with the P, it means plant zinc. So that's zinc contained in the plant, potassium contained in the plant, and then nitrogen contained in the plant, etc., etc. You can see that they are all closer to the annual. So closer to annual application of compost compared to the biennial, and then compared to the control letters. Okay. Then at the same time, but yes, there are some elements that are also associated to the biennial, a bit closer to the biennial. Like if you see magnesium, copper, etc., etc., they are closer to the biennial. If you look at the root length, you can see that it's closer to the biennial. It's closer to the biennial, the root length, because these they have to search. They don't have to be associated with the annual because the plants were happy in the annual, so they don't have to go deeper and deeper and deeper to search for mineral elements and so on and so forth. Then you look at plant height and yield, and can see that they are closer to the annual here. If you look at plant growth in general, looking at the shoot, plant growth in there, so plant height, the yield, and so on and so forth can be associated more to the biennial because they fall within the same this quadrant here. Then we also did metabolic analysis. So the metabolic analysis, the tricarboxylic acid analysis. So when we eat, the food goes through metabolic processes, okay, and several metabolic processes, and then ends up in this tricarboxylic acid cycle. And through the tricarboxylic acid cycle, energy is generated in humans. The same thing applies to plants. The same thing applies to animals. But these were measured in plants. So, so we can see that some of them, like glucose, for instance, glucose is a main product that gets into the starts the tricarboxylic acid cycle. So glucose and then pyruvic acid, and then it generates all this acid that you see, alpha ketoglutarate acid, and then the citric acid, succinic acid, fumaric acid, malic acid, etc., etc. They generate that. You can see that all of them, you can really associate that with compost. Okay, all of them can associate that with compost, except few of them like ketoglutarate acid. But you can see that the glucose, the fumaric, and so on and so forth, they all can all be closely associated with compost application compared to the control. So compost application is very, very important. This shows that this demonstrates that compost application is very important to plant health. It's very important to soil health. And therefore, at the end, we get our higher yield and get our good quality plant. We did not measure the quality of plant or report the quality of plant here, because in my previous presentations, we reported essential amino acids and some metabolic profiles of food produced in using compost, annual application of compost, and all that. The papers were published, so you can make reference to those papers as well. So in summary, our conclusion is that currently farmers are faced with many challenges related to soil health, which is made even worse by this sustained global climate change and the current global shortage and high cost of fertilizers. As we all know, in some countries, I visited Tanzania recently, I visited St. Lucia recently, and people will tell you that there, they don't even get fertilizer to use. So this year production is going to be very low, and I don't know how they're going to manage the food shortage, because they did not even get fertilizers to use these countries. And it's very, very expensive if you get at all from the black market. So this is a big, big issue. So why don't we use our resources, locally available resources, which is municipal solid waste, which is abundant everywhere? Why don't we use that to convert that to a useful product that we can use to sustain our soils and agriculture? So we affirm that annual application of municipal solid waste compost promote positive association between microbiome function and ecosystem services for the benefit of cropland, as we see in this presentation. And this will benefit farmers, and if implemented, that is, if compost, CQA tested compost is implemented and used as it should, will immensely benefit consumers with safe, nutritious, and affordable foods. So our notion that compost or municipal compost is not good, yes, sometimes you can have contaminants there, so it needs to be sorted so that the good compost is good feedstock is used for the composting. If we are able to do this, then we need not be scared of using municipal solid waste compost for producing food, because it's rich and it does the job. It does the job. So this is a summary, and as I've said, we published this paper recently. It was published 2 weeks ago, and the presentation is coming from this particular paper. So if you want to read more and know more, especially get the scientific understanding, maybe you have a question, a pertinent question that you want to get the scientific understanding why some of the results happen this way or that way. This is a presentation, I can't talk about it all, but it is all explained in this paper that you can use or you can read, make reference to. Thank you very much, and really appreciate your listening. Thanks.